The following was produced by researchers at the University of Georgia in the Warnell School of Forestry and Natural Resources. This demonstration video describes the harvesting of xylem and phloem tissue and subsequent RNA isolation from Pinus taeda, commonly known as Loblolly Pine, which is the single most important conifer species for forest products production in the United States. Once the tree is felled, in this case a 40-year-old tree, the main stem is cut into bolts that are approximately 50 centimeters in length. The bolt is then scored longitudinally with a chainsaw in several places to facilitate removal of the bark. Using a hammer and wood chisel, the bark is lifted up until it can be peeled away in sections. Next, the phloem tissue is removed by hand and is stripped from the inside of the bark sections and immediately immersed into liquid nitrogen. Once the bark has been removed, the thin layer of secondary xylem can be collected by scraping the bolt surface with a single-sided razor blade. The watery xylem tissue is immediately placed into liquid nitrogen. In this harvest, we also collected samples of reaction wood, commonly referred to as compression and opposite wood. Before cutting limbs from the main stem, the limb orientation is first marked with spray paint so that the areas containing the two tissue types, opposite wood on top of the limb and compression wood on the bottom, can be determined during bark removal. The limbs are first scored with a chainsaw along lines delineating compression and opposite wood, and bark removal is performed as with the main stem bolts, except that only bark in the region corresponding to the type of reaction wood being harvested